Okay, well, let's go and get started for the day. So in terms of announcements, so Quiz 11, I actually made an announcement on Wednesday that it was due today, but I had forgotten that I had just put it as Monday. So we'll just keep it as Monday, okay? So the due date for Quiz 11 will be Monday. We'll definitely have finished the material by the end of the day for that, okay? In fact, I hope to start to lecture 11 today as well, okay? But that'll be due Monday night, okay? We are gonna have a group work project not Monday next week, but either Wednesday or Friday of next week. So I'll probably, at this point in time, I'd probably say it's more likely to be Friday than it is Wednesday, the pace that we're going. So, but it could be possibly Wednesday depending on where I get to today and then Monday. Okay, so keep that in mind. And then finally, I posted a problem set yesterday, problem set 4, that's going to be due on the 3rd, May 3rd. Okay, so of course we're coming to the end of the semester, and we've got problem set 4, problem set 5, a couple quizzes left in that group work project. Okay, and then of course the final exam. Okay, final exam, by the way, is not cumulative. Okay, so not in a strict sense. So it'll only be over essentially lipid metabolism and amino acid metabolism. Okay, we're kind of in trouble at this point in time in terms of where we're at in the material. So you're going to start to see me hack away some things, meaning that there are going to be some things I'm just not going to cover. Okay, so probably fatty acid synthesis we won't spend a lot of time on. Okay, so unfortunately, but we have to get to amino acid metabolism in like at least 3 days on that subject. Okay, all right. So let's look at our riddle here. It said we all have one. We also say it to mean there's only one, and spelled quite differently. We use it to run, but wait, don't be confused. I'll give you some pity. When spoken out loud, it's the name of a city. Seoul, exactly. Seoul, Korea, the sole of your shoe, your everlasting soul, right? I mean, all these things, okay? Seoul, okay? All right. So, Wednesday, this would have been, well, wait, let's see if we can't open this. There we go. Wednesday, we finished our discussion on reverse cholesterol transport, and quite honestly, that then finished essentially this slide to the left, okay? So we have now gone over in their entirety the exogenous, the endogenous, and the reverse cholesterol transport pathways and seen how lipids and the challenges that they pose to our bodies in terms of transporting fairly hydrophobic substances, how those can be transported throughout a bloodstream, okay? And then absorbed by various tissues. What we want to do now is move into the cells and start to think about the biochemistry in a cell and how they're going to start to process lipids. Okay, in particular, we'll be thinking about fatty acids. Okay, so we'll start here at the synthesis of triacylglycerides. Okay, that's what we're going to start at. This would occur primarily in the liver or adipose tissue. Okay, so that's what we'll be thinking about at the beginning of the day. Once we're done with that, we'll shift gears and we'll move from there into then other tissues and what they will do in terms of fatty acid breakdown, what we will call beta oxidation. That will be lecture 11. Okay, so those will be our two major topics today and more than likely Monday. Okay, so let's start with this slide here, and specifically in the liver, you can think about dietary carbohydrates, dietary proteins. Those will be broken down, of course, into their individual subunits or monomers, and those are going to funnel into this thing called acetyl-CoA. Okay, acetyl-CoA in a liver is going to be converted primarily to fatty acids. Okay, so insulin is going to be a signal to make fatty acids from acetyl-CoA. Again, a pathway that probably we won't spend a lot of time talking about, but we'll try and get to some of it. But that'll make fatty acids, and then from fatty acids, triacylglycerides will be made. Okay. So triacylglycerides can also be and will also be made in adipose tissue. Okay. So if you go back and look at your slides, both the exogenous pathway and the endogenous pathway, by the way, this could happen in the small intestine as well, okay, triacylglyceride synthesis. All right, but if you go back and look at those pathways, you will see that I said, okay, well, we'll just make triacylglycerides, and then didn't give you really a lot of information. So that's what I want to do is fill in some of those missing gaps there, okay? And there's really going to be two steps to this process. The first step is going to be make acyl-CoA, okay, from free fatty acids. And then two, we have to make glycerol 3-phosphate. Okay, and I said there's two steps. Really, there would be a step after that. Combine these to make triacylglycerides. Okay, and that would be step really number 3. So that's how we're going to order this. Talk about acyl-CoA production, glycerol 3-phosphate production, and then combining those two to make the triacylglycerides. Okay, and then of course in a liver cell in particular, a liver cell will then package that in a VLDL and send that off. A small intestine cell is going to package that in a chylomicron. Okay, adipose tissue is just going to store it in a fat droplet. Okay, and then it will actually break it down and ship it out when the energy need is needed. Okay. All right, so let's move on and let's talk about this first step here, okay? Sources of acyl-CoA. So if you're going back on my last slide, this is the reaction that's going to be required to make acyl-CoA. Now, we are putting this in the context of a liver cell, but understand that pretty much any cell that takes up free fatty acids, this is going to happen. Okay, free fatty acids are very rarely, if ever, found as just that, a free fatty acid in a cellular environment. They, generally speaking, are quote-unquote carried by a coenzyme A molecule. Okay, and to produce the acyl-CoA, this is the reaction that's needed. There's actually 2 steps, but 1 enzyme that's carrying out these steps. So the name of the enzyme is listed here, fatty acyl-CoA synthetase. Okay. And it's showing both of these steps. Okay, the very first step here would be essentially the denylation of the acyl group. Okay, so the acyl group, just so we know what we're talking about, that's our acyl group. That's going to nucleophilically attack the last phosphate, gamma phosphate, on ATP. Okay, nucleophilic substitution will lead to then essentially this, an adenylated fatty acyl group, okay? And this is just an acyl group with an adenylate molecule attached to it, okay? Adenylate, this would be essentially AMP, right? So it's adenosine monophosphate, but we'll call it adenylate. Okay? And that's the first step of this. Now, that then breaks off pyrophosphate. And pyrophosphate, anytime it's released in a cellular environment, there's an enzyme called pyrophosphatase that's fairly efficient that will break that up into inorganic phosphate. That will actually release some energy as well. Now, step 2 then, we have this adenylate or adenylated acyl group. Okay, is going to be another nucleophilic substitution. The coenzyme A is going to, shown here, is going to nucleophilically attack our acyl group. It's going to displace the adenylate group. The AMP is going to come off. And then you'll have here fatty acyl-CoA. Okay. A fairly significant amount of energy is stored in the acyl-CoA molecule. Okay, acetyl-CoA, if you remember, I talked about that as having energy stored from a redox reaction produced from what we put it in the context of would be the pyruvate dehydrogenase complex reaction. Fatty acyl group has energy stored from essentially the hydrolysis of ATP, In this case, though, it's really removing, it's breaking ATP down to AMP and 2 inorganic phosphates. Okay, so quite a bit of energy stored in this. Okay, because of this thioester here. Okay. And this stores energy. Now, something to recognize just while we're on this topic of energy too, this is the first enzyme that we have encountered in this class that uses ATP and doesn't make ADP from it, but makes AMP. Okay. When an enzyme does this, we will say that the enzyme utilizes 2 ATP equivalents, though it's 1 ATP molecule that it's using. Okay, so yeah, some people were like, well, that doesn't make any sense. How can it be 2 ATP equivalent if it's only 1 ATP molecule? Okay, well, the reason is this. Anytime an enzyme breaks down ATP to ADP, that ADP can be combined with another ADP to make ATP plus AMP. Okay? And there's an enzyme in all of your cells, okay, called the adenylate kinase that does that reaction. Okay? So if you look at hexokinase or PFK1, those would be examples of enzymes that we saw that hydrolyze ATP to ADP. They'll split off this ADP molecule that then can go on and make an additional ATP. Okay? Well, in this case, the fatty acyl-CoA synthetase, it doesn't produce ADP, it produces AMP, right? So the reaction that we see over here for this enzyme is not available. And so then we say that it uses 1 ATP directly, but then because it uses the additional phosphate, it loses the capacity to make another ATP molecule. So we'll call it 2 ATP equivalents, okay? So when we get to counting, like we'll talk about this later on because we'll do this with fatty acids breakdown as well. We have to make an acyl-CoA there to break down fatty acids. We have to remove, or we will, in our accounting, we have to remove 2 ATP equivalents, okay, at the beginning of the pathway, okay, to kind of get the pathway going, okay? All right. Questions on that? This will be the very first step of beta oxidation too, okay? Getting fatty acids prepared to go through energy harvesting, okay? All right, now let's go here. This would be, if you come back here, right, we just did this. Make acyl-CoA. We need to go now to make glycerol 3-phosphate, okay? So this would be step number 2 here. And there are actually a number of ways to do this. And we're going to look at all 3 of them. Okay, so the first one is really a very direct means of doing this in terms of what we've talked about in this class. So this would be number 1 source. Start with glucose, break it down through glycolysis to dihydroxyacetone phosphate, DHAP. Some of you say that sounds vaguely familiar. Good. It should, okay? It's the one of 2 products of the 4th step in the glycolytic pathway. So glycolysis at the aldolase reaction splits fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So that's where that's coming from, okay? Now, the next step is not found in glycolysis. It's just an enzyme, a single enzyme, glycerol 3-phosphate dehydrogenase, that will take dihydroxyacetone phosphate and make glycerol 3-phosphate. Okay. Not glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is an aldehyde. Glycerol 3-phosphate is an alcohol, or has 2 alcohol functional groups. Okay. Now, some of you say, well, that sounds vaguely familiar. Glycerol 3-phosphate dehydrogenase. Yes, it's part of the glycerol 3-phosphate shuttle. Okay. This is not going to complete the shuttle, but that's the same enzyme. Okay. So that's one of the sources. Okay, and that can happen in multiple tissue types. Okay. Another one of the sources, so this would be source number 2, and talk about straightforward, okay, would be to start with glycerol. Why not? Okay, your body has, of course, glycerol in it. Okay, by the breakdown of triacylglycerides, there's going to be a supply of glycerol. That glycerol can be absorbed by the liver or by the kidney, and this enzyme, glycerol kinase, liver primarily or kidney, okay, can directly phosphorylate that glycerol molecule, the glycerol 3-phosphate. We actually encountered that before. We've already talked about it. Okay, so that's a very straightforward process, just a single step. And then finally, source number 3 would be a process called glyceroneogenesis, which is not fun to say, glyceroneogenesis. Okay, so that is going to happen primarily in adipose tissue. Okay. What's interesting about adipose tissue is this. It is not a gluconeogenic tissue. Okay, so liver is, of course, kidney is, but adipose is not, and yet the adipose tissue expresses pyruvate carboxylase and PepC-K. These are the first 2 steps in gluconeogenesis. These cells lack the glucose 6-phosphatase, and I'm pretty sure they lack FBPase1, but I'm not positive about that. But they do lack glucose 6-phosphatase. But they have these 2 enzymes in them. Okay, now why would they have these enzymes in them? Well, it's for this process primarily of glyceroneogenesis. And pyruvate carboxylase, as we've already talked about, this is just some pathway that we've already looked at. Okay, you would take pyruvate, carboxylate it to oxoacetate. Oxoacetate is going to be decarboxylated to phosphoenolpyruvate by PepC-K. Both of those processes require ATP or an ATP equivalent, remember. Okay, and then multiple steps that we've already mentioned, this would be glycolysis in the reverse, okay, will make dihydroxyacetone phosphate. Okay, the enzyme that we have over here is also found in this tissue, glycerol 3-phosphate, dehydrogenase, and that will make then glycerol 3-phosphate. Okay. Yeah.
These pathways make glycerol at the same time, or would one happen?
Yeah, okay, so the question is, can these pathways make glycerol at the same time? You would have to have a phosphatase expressed to remove this phosphate to make glycerol. So, and there is a phosphatase like that. I'm not entirely sure that there would be a high flux or demand for that, simply because there's not really anything that glycerol, just straight glycerol, is used for. Okay, usually it would be the opposite direction. We want to take glycerol, add the phosphate to it so that we can make the triacylglycerides out of it. Okay, the liver can and other tissues can break down glycerol, feed it into the glycolytic pathway for energy harvesting, but it has to get it to dihydroxyacetone phosphate or glyceraldehyde 3-phosphate or one of the other aspects of it. So usually you don't see that production of glycerol. You'd have to have a phosphatase to do it. I don't know if that phosphatase exists. Okay, so other questions? No? Okay, so that's number 2 up here, right? So we've done this now. So now we just need to make the triacylglycerides. Combine those 2 and make the triacylglycerides. Okay, so let's go here. We've already done that. We don't need to do that. We just come over here, okay, and we take the glycerol 3-phosphate that was made in one of those processes that we just looked at, and just add simply, so let me write this down, acyl-CoA to it. Okay, coenzyme A comes off of it. Okay. That would make what would be considered a monoacylglyceride, right? MAG. Do that again. Okay. Again, acyl-CoA. And that would make, it says phosphatidic acid, something that we could also call diacylglyceride. Okay. It has a phosphate on it. So phosphatidic acid is really more appropriate, okay, because you haven't removed the phosphate yet. But the enzyme that does this is acyltransferase. Okay, it's just transferring an acyl group from coenzyme to the glycerol group. Now, one more step to make triacylglycerides. You take that phosphatidic acid from our last slide, and you break off the phosphate group using a phosphatase. Okay? And then you again add another acyl-CoA. Okay? And that is done again by acyltransferase. That will make triacylglyceride, or TAG. Now you'll notice here that you could also go in the opposite direction, right? Once you have the phosphatidic acid, okay, you can make from that membrane lipids, okay? Right. And of course, you know, you're looking at phospholipids here. There will be a number of enzymes that can add some head group to that phosphatidic acid. Okay, so then they can make phosphatidylcholine or phosphatidylserine or phosphatidylinositol or something to that effect. Okay. And then this, having completed that process, this will hopefully be packaged in a VLDL if this is being done in the liver, and off it goes. Okay. Or if it's done in adipose tissue, it'd be right into a fat droplet. Or in the small intestine, a chylomicron. Okay. And those would be the three main tissue types that are going to be doing this. Questions on that? Okay. This would be happening in a fed state, generally speaking. Okay, so after a meal. Yeah, question?
What is the, it says R squared, is that just 2 R groups? Or?
Oh, yeah. Okay, so this right here. Yeah. So R1 would be the acyl chain. Okay, whatever it is, usually it's between 16 to 24 carbons long. Okay, R2 would be just the second one. Again, a 16 to 24 carbon chain, with depending on it could be saturated, it could be polyunsaturated, something like that. Yeah. Other questions? Okay. Let's see here. Do I have? I'm not going to ask that question, okay? Maybe get to another question. Let's do this, though. Let's step into one special topic real quickly here. I don't want to spend a lot of time on this, but I do. It's interesting, okay? Because we've talked about diabetes now. We talked about diabetes, of course, before spring break when we were talking about carbohydrate metabolism, in particular type 2 diabetes, and I referenced the fact that there is this correlation between someone's BMI, right, body mass index, and their risk at contracting type 2 diabetes. All right, I said that 90% of individuals with type 2 diabetes have, 90% of people, with type 2 diabetes are obese, okay, from a clinical standpoint. 10% are not, but there's obviously a very strong correlation there. Okay, there's a genetic component that we won't look at here. But I do want to go ahead and talk about, well, why would there be a higher risk for an obese individual to contract type 2 diabetes? Type 2 diabetes, remember, the hallmark of that is essentially insulin resistance. Okay, that's one feature of the pathology of this disease, is that these individuals will have sufficient insulin supply, but the insulin is not effective in terms of bringing down glucose levels in the bloodstream. Okay, and what you're looking at here would be a possible explanation for that. Okay, and there's been a lot of research done into this, so this is a pretty well supported slide. What you'd have here, this would be, right, what would be considered a healthy body mass index, what might be considered lean. So thiacylglycerides, what they're trying to communicate here, are equal to the dietary intake of lipids is equal to the dietary or the energetic needs for those. Okay, so you're not gaining weight in that regard. Of course, everything to the right would be the potential that lipid intake is greater than the need, okay, for the energy production, and therefore what's going to happen is that those lipids, as they're processed in the liver to VLDLs, the VLDLs go ship those out. The lipids are not used, okay, and those will be, generally speaking, absorbed by adipose tissue, which is, you know, pretty much ubiquitous in the body, and that will then lead to larger adipocytes. Okay. Now, as those adipocytes start to get larger, okay, that can be problematic to the adipocytes. Okay, so you, generally speaking, you don't make more adipocytes; they just grow bigger, right? And by growing bigger, that can lead to an inflammatory state, okay, or lead to essentially inflammation, and that's what step number 3 is essentially saying here, okay. The lipids are becoming cytotoxic to these cells. Those cells will start to release a factor called, it's actually listed here, macrophage chemotaxis protein, okay, that will elicit an immune response. Okay, macrophages will come to these sites, okay, and that's what's shown here. These are macrophages, okay, apparently the high resolution structure of them, okay. Those will start to release a factor called TNF alpha, tumor necrosis factor alpha, okay. And what that will do, the tumor necrosis factor alpha, will actually be a signal to the lipid or the adipocytes, you know what, you've got enough lipids, just get rid of it, okay? So what will start to happen in this case, and you see this here, fatty acids will start to be released from adipocytes even when signals in the body are not indicating that there's an energy need, okay? A healthy adipocyte will release fatty acids when told to do so by glucagon. Glucagon being a signal that energy levels are low, you're in a fasted state. Okay, then they will start to release fatty acids, and then those fatty acids will be used for energy needs. In this case, the lipids are not being released by the adipocytes because of glucagon and energy need, but they're being released because the cells themselves are being harmed by the lipids. And so they're getting rid of them, they're expelled, putting them out into the bloodstream. Those will then be absorbed by other tissues, the liver and everything else. Okay, but they have to go somewhere. Signals in adipocytes are released, don't take in. Okay, and so what will start to happen is that tissues that are not really designed to store fats will start to store fat. Okay, and that's what you're essentially seeing here. Okay, and it's called ectopic storage of fat. Okay, what we have essentially fat storage or lipid storage in this case is skeletal muscle in other tissues. Skeletal muscle would be the big one, okay? That if skeletal muscle is having to store more than it would normally do, and it doesn't store very much lipids, then that can become toxic to those cell types. Right? So, we've got fat in the muscle. That's the take home of this. Okay. What does that do? Well, it does lots of things, but let me just show you this. This is a figure that's not in your book. This can get real complicated real fast because it's insulin signaling, and insulin signaling is complicated. Okay. Let me just put a box around these 3 different proteins. Okay. PKC, protein kinase C, JNK, okay, JNK, which is a kinase, and then this phosphatase, PP2A. There's lots of research that demonstrated that what's called ectopic fat storage, or fat in the muscle, will actually activate those factors, okay, those different proteins. This is where it can become kind of confusing, but so I don't want to try and muddy the waters here, but let's just say this, okay? These are going to inhibit aspects of insulin signaling, okay? These 2, PKC and JNK, are going to phosphorylate insulin receptor substrate in sites that will inhibit it rather than activate it. Okay. So insulin receptor substrate is something that we talked about, is activated by the tyrosine kinase receptor when insulin binds. Okay. And it's activated through phosphorylation. However, the IRS, insulin receptor substrate, has lots of different phosphorylation sites. Some of them activate it, some of them deactivate it. These are deactivating phosphorylations. That will halt insulin signaling at its, really, the forefront of the signal. So even if insulin is present, you've got these competing signals that are going to reduce the overall effect of insulin bound to the receptor. That's going to make then that cell that has this going on insulin resistant. Okay? Same thing with, well, it's not really the same thing with PP2A. PP2A is a phosphatase. And if signals get past this, PI3 kinase that makes PIP3 that then will recruit AKT, AKT will start to be phosphorylated. PP2A, when it's activated, will actually be actively dephosphorylating AKT. So even if AKT is getting phosphorylated, which is going to become active in response to insulin signaling, PP2A, which is being activated by all these fats, is going to actually downregulate that. And so as a whole, these factors are going to short circuit insulin signaling even if insulin is there, right? And they're activated, and we're not talking about how, but they're activated by this abnormal amount of fat that would be found in these cells. Okay. And that can then, of course, lead to what the condition that we call type 2 diabetes, if it happens on a massive scale. Okay. Okay. Questions on that? Yeah. Yeah, so that's something that, so I'd have to go look further into that. And I'll try and remember to post a paper, a review. This is actually, I think, a review that was published about 4 years ago, so there's probably more information on this than what I have. My suspicion, though, is that there's other downstream factors, that are activating or inhibiting these types of things that are binding to the lipids. So I don't know, is the short answer. Yeah. The short and most honest answer. Okay. Other questions? I will say this, ceramide is really important in this process. Okay, a type of lipid. So, and I have to go back and look at it, what causes it. Questions? Other questions? Okay, all right, so let's do this. Let's skip this slide. Okay, we might come back to it. This is how lipids will be mobilized in a fed state. So that would be the healthy aspect of this, right? I'm not in a fed state, but in a fasted state. So lipids that are stored in, of course, adipocytes and glucagon can activate that whole process. What is being shown here is essentially how glucagon will activate mobilization of fatty acids out of the lipid droplet. Free fatty acids will leave these and be bound by this protein albumin, and then they'll be taken out to cell types, okay? I don't want to go over it, though, right now because we're crunched for time, okay? All right, so let's do this. Let's go to lecture 11, Okay, and in lecture 11, again, we're going to stay in the cell. Fatty acids are coming into the cell. Okay, certainly fatty acids are a primary source of energy for lots of different cell types. In fact, they're the preferred source of energy for lots of different cell types. So what we want to do is look at then how are lipids going to be catabolized. A portion of this lecture would also be fatty acid synthesis, which we will have probably an abbreviated discussion on that. Okay. What I do want to spend time discussing in some detail is the carnitine shuttle beta oxidation and ketone body production. Okay. So we'll talk about those. So let's start here at the carnitine shuttle. And the carnitine shuttle, before we get to this slide, is just the means to get a fatty acid into the mitochondria. Okay, so for energy purposes, the fatty acid is going to go through beta oxidation, and all of the enzymes for beta oxidation are found in the matrix of the mitochondria. Okay. However, acyl-CoA, there's not a transporter that will directly transport acyl-CoA into the matrix of the mitochondria. So we have the shuttle system set up, and you say, oh no, because you've got this idea of the malate aspartate shuttle like churning in your mind, and that's, yeah, I mean, that's good that you think that. Okay, there are these shuttles that get things into the matrix of the mitochondria, and this is one of them. Okay, we've already gone over this slide, so we just went over this actually in the production of triacylglycerides. This is how acyl-CoAs are made. So that would be step 1 of the carnitine shuttle. Okay, step 2 and 3 are shown in this, I think, somewhat effective slide. Okay, so quickly here, this would be carnitine, just so you have some idea what we mean by carnitine shuttle. It's just a small molecule. Okay, when we talk about an acylcarnitine, it will have the acyl group or the fatty acid group attached to that carbon there and that oxygen. Okay. All right. So this would be the way I just drew that. That would be acylcarnitine. Okay. To make acylcarnitine, you start with acyl-CoA. Okay, and essentially do a substitution of the CoA group with carnitine. Okay, that's done by this enzyme carnitine acyltransferase, or CAT1. CAT1 is the rate-limiting step of beta oxidation. It's not really even part of beta oxidation. Okay. But the primary means of regulating lipid catabolism is by regulating CAT1. If you inhibit CAT1, you can't move fatty acids into the matrix of mitochondria, and they can't be broken down. Okay? Now, once you get acylcarnitine, then it will move into the matrix of the mitochondria through a transporter. That transporter will move carnitine out. Not acylcarnitine, but just carnitine. So it's an acylcarnitine antiporter. Okay. Acylcarnitine goes in, carnitine comes out. And then you have CAT2. Carnitine acyltransferase 2. Okay. That will then take that acylcarnitine, make the carnitine and a coenzyme, and make acyl-CoA. So now we're back to acyl-CoA. Okay. Now it's ready. Okay, you can go through beta oxidation. It's in the matrix of the mitochondria. Having said that, to start this whole process, remember, coming back here, to make the acyl-CoA group, it costs 2 ATP. All right, so this again, it costs 2 ATP equivalents. Okay, so when we start to think about, well, numbers of ATP per fatty acid like we did with glucose, we have to subtract 2 ATP at the top of this. Okay? This would be equivalent to, if you want to think about it this way, like an energy investment stage of glycolysis. Okay? So. All right. Now, beta oxidation. I don't want to spend a lot of time on the introduction here. Okay? Beta oxidation is going to do this. Okay? It's going to make acetyl-CoA, and it's going to make a whole lot of NADH and FADH2. Acetyl-CoA can, of course, feed into the citric acid cycle and make more NADH and FADH2. Now, they're giving numbers here of electrons, okay, and acetyl-CoAs, and that's based off of a 16-carbon acyl chain, okay, which we'll talk about the breakdown of a 16-carbon acyl chain. Okay. And then, of course, those electrons are going to enter into oxidative phosphorylation. And a whole lot of ATP is going to be made, a lot more than you would see for, let's say, carbohydrates. Okay? Now, I do want to ask one question here. So that's our introduction to beta oxidation. Let me ask one question, though, okay, for today, just to make sure we're all on the same page with lipid, in particular, fatty acid nomenclature. Okay, so if I were to say that we are interested in the lipid 18:1D9, D is delta and 9 would be a superscript to that, so it'd be 18:1 delta superscript 9. What does this tell you about the lipid? Let me be more specific. How many carbons is it? Is it saturated? Is it unsaturated? Okay, what do these terms mean? Okay. All right, so 18:1 delta 9. Okay, on your marks, get set, go. What do you think? Okay, how many carbons? Is it saturated or unsaturated? And you've got a minute 30 left. You can talk to your neighbor about that if you want. Okay. Yeah. Is not? You got it? Okay, good. I was hoping so. Whew. Okay, you've got a minute left, or less than a minute left. About 20 seconds left. So submit an answer. How many carbons is it saturated or unsaturated? All right, 5 seconds. Okay, here we go. Let's check this out. Okay, so 18 carbons saturated, 18 carbons unsaturated, 18 carbons unsaturated? Question mark. Okay, monosaturated, unsaturated, 18, double bond at carbon 9. Okay, good. I don't know. It's a lipid. Very good. All right. Baseball. Yeah, well, that is baseball season. So you wanted to hear about the Rockies. Okay, so fine. The Rockies are continuing to win. They're in upward progression. Okay, it's a good thing right now. I feel optimistic that I can make it through the rest of the summer. Okay, well, that's a good thing for me. I'm feeling a little bit like I can breathe now. Okay, they're not losing as much. Okay, go Rockies, exactly right. Okay, now let's do this, okay? So lots of you are hitting around the right answer. In fact, if I take the combination of a couple of them, there's, like for instance, this one, 18 carbons unsaturated. There was one that said 9 carbons, no, let's see. It said a double bond at the 9th carbon, and that's right. And then this person, I'm so sorry for you. I really do. I feel bad for you. It's okay. You're going to make it through today and the weekend. Okay. I can't say anything more than that, but today. Okay, so let's look at this. I introduced this just because it's a good way to talk about nomenclature. Nomenclature of fatty acids can get a little bit confusing because there's lots of different conventions. We're going to use the convention that's listed here, okay? This is our what I was saying, 18:1 delta upper or superscript 9. What that means is there's 18 carbons, so that's right. Okay, so if you count that, there's 18 carbons there. And then it's unsaturated because it has a double bond in it. So saturated means no double bonds. Unsaturated means that there's a double bond, and the double bond begins at the 9th carbon. Okay, so yes?
Would this notation be different if the double bond was trans versus cis?
Yeah, so the question would be, would the notation be different if it was trans versus cis? You would actually put that, you would have to label cis or trans in this, okay? It's not showing this, cis or trans, but you'd put the trans or cis somewhere, okay, to indicate that. I will say this, so when you start to look at naturally occurring fatty acids, none of them are trans, okay, so they're cis. All right, so we don't deal with a lot of trans double bonds, okay? So that's how you would label that. Now, to go down, below here, right, just to make sure that we're all on the same page, this is a 20 carbon fatty acid that has 5 double bonds in it, starting at the 5th, the 8th, the 11th, the 14th, and the 17th, okay, carbons, all right? That would be what we call polyunsaturated, okay? Now, the other aspect of this that's very important for our discussion, because I've mentioned several times that we're going to be going over this pathway called beta oxidation. So the second carbon is referred to as the alpha carbon, okay, the carbon just adjacent to the carboxylic acid, which makes the third carbon beta. Okay, and that's the one, and that's true on any of these, that's the one that's going to be oxidized as we go through this pathway. Okay? That's why we call it beta oxidation. Now, the other thing that you may have heard of are omega fatty acids. Okay, so you may have heard of like omega-6 fatty acids or omega-3 and omega-6 fatty acids. And they're essential fatty acids, and they're healthy, and there's lots of health benefits to them. Omega, that convention, okay, is going to name this based off of N minus number, okay, where N is the number of carbons. Okay, and then number, okay, is going to be position of the double bond. Okay, so when you get to omega-6, for instance, 20, if we're coming here, because this is an example of this, 20 is our number of carbons there. There is a double bond at the 14th carbon, minus 14 is 6. So this is what would be considered an omega-6 fatty acid. Okay. It would also have one at 17, so it would be an omega-3 and 6 fatty acid. Okay. So that's where that convention comes from. Okay. We won't be dealing a lot with omega amino acids in this class because we're not going to get into eicosanoids. I wanted to, but we're not going to. But if you hear about omega amino acids, not amino acids, fatty acids, I'm sorry, and that's what it means. Did I say amino acids like 7 times, or is that, okay. Let's stop there while I'm ahead, right? So we'll start beta oxidation on Monday, okay? All right.